Alright guys, welcome back. We're going to go ahead and get started here. I do want to address a few things. If you haven't checked your email, there was a letter sent out to you and your parents, I believe, regarding grades. So if you have a question regarding that, go ahead and send me an email. But I'm just going to give you the nutshell of how it works for this class. Being this is a year-long course, it has always been this way that we average your first semester and second semester grades together. So we're going to take your first semester grade percentage-wise and average that with your second semester grade that we have so far. Okay, that will then determine your letter grade. All right, if you have any questions on that, like I said, don't hesitate to email and ask questions on that. All right, so we're going to pick up right where we left off last time, talking about the cell cycle and mitosis. Last time we did mention that there was interphase, which is the longest phase of the cell's life, and that's where the cell does most of its normal work for you. There are three subphases, the G1, S, and G2. Mitosis and cytokinesis are also a part of this, and we kind of mentioned mitosis a little bit, and we also touched base on cytokinesis. So let's go into a little more detail regarding mitosis. We said there were four phases: prophase, metaphase, anaphase, and telophase. The first one we talked about in prophase, and we said that prophase is the very first phase where the nuclear membrane will break down. The chromosomes are now visible. They are replicated in their sister chromatid form. We know that they've replicated in the S subphase of interphase. And then we see these mTOCs, or microtubule organizing centers. And that's, again, everything from spindle fibers to centrioles, all that good stuff in helping us divide our DNA equally into two new cells. All right, we got to metaphase where they line up along the equator. And again, this is for logistic reasons, so that both the sister chromatids arrive at the poles at the same time. Okay, so metaphase where they line up along the equator of the cell. And again, how they moving around and stuff like that to the center, that's your motor proteins. The little motor proteins that we talked about in the previous chapter. They're being powered by ATP, are moving those chromosomes with the help of the spindle fibers towards the center. We're also going to use motor proteins to help pull those things apart and towards the poles as well. Anaphase, you can see that the sister chromatids have pulled apart. Now we have the individual pieces going in opposite directions. So this is the exact same copy as this one here, and this one, same copy here. So each new cell will have the exact same DNA. In telophase, we have two new identical cells starting to form. We see that the nuclear membrane is starting to reform. We have our chromosomes there. They're going to eventually relax or decondense down into their loosey-goosey form. So that way they can do transcription and translation to make the valuable proteins that you need for everyday life. Okay, so are the newly formed cells haploid or diploid? Haploid, as we talked about, means half the genetic information. Diploid means a full set. So do these cells have half the genetic information, or do they have a full set of DNA? The correct answer is full set. All right, because these cells are 2N or represent like skin cells, heart cells, things like that. The only time they're going to be haploid is when they are egg or sperm cells. Okay, so we got diploid cells right here. Okay, so cytokinesis is not a phase. Okay, keep that in mind. It's not a phase. It is a process that happens at the end of mitosis, where the cell will start to pinch inward, making this contractile ring or cleavage furrow, and which is basically just a pinching in of the cytoplasm. It's going to divide the cytoplasm, the contents of the cytoplasm, which may include things like mitochondria as well. So as such, this cleavage furrow will start to shrink down, the contractile ring shrink down. Think of it like a balloon, like making a balloon animal. You start to pinch in on it. It makes that gap between the two pockets of air smaller and smaller and smaller until eventually it just presses together and they just pop right apart. And you have two new identical daughter cells that are, again, diploid or 2N. Plant cells do the same process. They have to grow. They have to add structure. One thing different about plants versus animals, though, is that plants have a cell wall. We, as animals, do not have a cell wall. So they have an extra step they must complete after mitosis is done. So they have the cell plate, which grows outward and to become a new cell wall, dividing the newly formed cells. Vesicles deposit materials in the middle to build that cell wall up. Okay, so they have to do one extra step. Otherwise, if they didn't, they would basically just be one giant cell wall and a bunch of cells inside there. And again, they'd probably be limited to their size based on that little cramped space. So it's a good thing that they build a new cell wall. This gives each new cell the support and strength that it needs in order to help maintain that turgid pressure when we talk about water, the plant's uptake, so they don't explode. All right. So these are very important checkpoints. So they divide these out, yes, in little bitty phases, processes, and things like that. And you're like, okay, whoop-de-doo. Well, each one has a specific set of goals or benchmarks they have to meet in order to progress to the next one. If they are not complete or not done, then we have a problem. Okay, so even interphase, as you can see, has the G1, G2, and the meiotic phase or mitosis. But they must make sure everything has been properly made or formed before proceeding to the next step. If not, you get cancer cells. Yay, that's bad. So nobody wants cancer. Cancer is a natural part of life sometimes. Just because cells sometimes mess up, mutate, and whatnot, it is a chance that yes, you've had a cancer cell or two in your body before, but your body has recognized it and killed it off. So yes, your body can repair itself and get rid of cancer by itself. Sometimes certain cancers become very aggressive and they multiply quickly, and that's when it becomes a problem. So not always do we catch those things in time. And as you get older, yes, your chances increase just because your body starts to wear out. Okay, so cells bypass these checkpoints that ensure the quality of the cell, but they can still grow and they become cancerous. They no longer work for you, they work for themselves. That's what usually creates this lump or mass within the body's tissue. That's usually what doctors have to go in and cut out, or they use the chemo and radiation to shrink the size of those cancer cells. So we are starting to find some really cool treatments and possibly cures one day for cancer out there. Some really cool information out there. I think we talked about this maybe at the beginning of the year. There was a Stanford news release that they actually did a treatment on mice, and it was like 98% effective. That it would treat a specific type of cancer, not all cancers, but a specific type. But they did it in mice, and it was very, very effective at getting rid of it. So they were soon to start human trials, which hopefully we'll start to see some publications on that as well. But if you haven't read this book before, The Immortal Life of Henrietta Lacks, it's a very cool read, especially since you've got some time off here. If you want a cool read about her, this woman, essentially in a nutshell, she had cancer, and they took her cells and they continued to grow them in a lab for many years. Turns out there was a lot of moral reasoning behind this, I guess, like dilemma, I guess you could call it, because her cells were continually being used and sold in kits and stuff like that. So they were kind of doing some weird stuff, but her family was unaware of this, and she'd passed away, so she was unaware of this too. So really cool read if you want to give it a look. Talk about cancer and some moral dilemmas as well. Okay, so let's jump into meiosis. So there's mitosis, which is going to produce your diploid cells. Then there's meiosis, which is going to produce haploid cells. And those are again for egg and sperm cells, which are collectively known as gametes. So daughter cells produced are not identical. These are unique cells in every single way. And yes, you produce these. Okay, this is getting a plethora of different combinations using your DNA to give to potentially your offspring one day. So one replication of the DNA, but two divisions of the cells. So mitosis only has one division of the cell. Okay, in meiosis you have the division 1, which separates the homologous chromosomes, and you have division 2, which separates the sister chromatids. And we talked about homologous versus sister chromatids in the previous part of this lecture. So if you forgot what that is, go back and see lecture part 1 of chapter 11. So just to give you the basics of meiosis, you have the first division, and we go through the same thing like we would in mitosis. We call them prophase, metaphase, anaphase, and telophase because many of the processes are exactly the same with some slight differences. We do number them though, prophase 1 and prophase 2. Why? To represent the round of division that you're on. So if it just said prophase, that would be mitosis. If it said prophase 1, that's meiosis. Prophase 2, still meiosis, but the second round of division. Okay, so as you look, you can see we have a number of things here. This is the second round. You've got your homologous chromosomes, which have already separated here. So the big one are in two different cells, small one and small one in different cells. Here we're going to see the sister chromatids separate in Interphase 2, and we're going to make 4 identical haploid cells, or 4 not identical haploid cells. Okay, and you're thinking, how are they not identical? They all have blue and red. If you look closely, this is a blue and red, and that's blue with some red, and that's red with blue. So they've done this process of crossing over, which we'll mention here in a few. Okay, so let's talk about meiosis, jump right into it. So you've got prophase 1, which is the very first phase. The chromosomes have been replicated, they are visible, the nuclear membrane has dissolved, we no longer see that. Homologous chromosomes synapse together to form a tetrad, and the word tetra actually means 4. So if you have 2 homologous chromosomes, one that you inherit from mom, one you got from dad, and they've both replicated in their X form, all right, you put them together, that is 1, 2, 3, 4 copies of chromosomes, as you can see right here. So 1, 2, 3, 4. So one's from mom, one from dad. And in that process, when they synapse together, they will do what is called crossing over, where they exchange some chromosomal segments between them. So some genes, essentially. So you're taking the chromosome that dad gave you and the one that mom gave you, and you're taking it like a deck of cards and shuffling them together and then dealing them back out. 52 here, 52 there. So they are both full set of full decks. Now, as you look through the deck, you've got some that are red and some that are blue. You got over here some that are red and some that are blue, which means you have recombinized your parents' DNA inside you, and you're going to give that to your offspring. That's pretty crazy when you think about it. Yeah. So that's how sexual reproduction works, is the recombination of DNA, ensuring that you get, hopefully, the best of both worlds from the parents. So in metaphase 1, same thing happens. They line up along the equator. However, there is one key difference. The homologous chromosomes are stacked upon each other like this. They're not all completely lined up along the equator. The homologous chromosomes are stacked. So in the case for your cells, you would have 23 right here and another 23 on top, stacked together. All right, so that's one key difference between meiosis and mitosis. In anaphase 1, the homologous chromosomes separate. So we're not separating the sister chromatids, only the homologous chromosomes are pulling apart. In telophase 1, the homologous chromosomes arrive at the poles. At this point, neither cell is identical. So they're unique in every way possible right now, just because of that crossing over. And again, this chromosome you got from dad, this one you got from mom. So there's already going to be some slight differences in the genetic code that they gave you. Okay? So now you're creating unique gametes, egg or sperm cells. So the second round of meiosis, or second division, is very, very, very similar to mitosis. Spindle fibers form. These chromosomes are hanging out there. The nuclear membrane's gone. Metaphase 2. Now they line up along the equator. And in anaphase 2, now the sister chromatids are going to be pulled apart. Telophase 2, the nuclear membrane starts to reform around the DNA. And then you have cytokinesis with 4 haploid cells, or N cells, that are unique. So again, we've got through meiosis 1, meiosis 2 here. You can see the picture again. Sister chromatids separating, and we have 4 unique haploid cells. Alright, so importance of meiosis provides variation. This is very important. So that allows for sexual reproduction, recombination of gametes during fertilization. Crossing over also produces new combinations of chromosomes, and it keeps the chromosomal number constant, because as we know, polyploidy in humans is lethal. So it is important that you get the right amount of chromosomes to ensure the survival of the offspring. Okay, and you're getting one set from mom, one set from dad, and that's kind of like getting a backup set just in case you have faulty genes. So it's a good thing that you have an extra set, essentially, okay, because you never know what you're going to get. But this is very important for unique individuals within the population, because if you just cloned yourselves, well, you'd never have any new traits theoretically coming into being unless they were just random mutations. But when you cross 2 individuals together, you're getting a different combination of traits. That's why you and your siblings don't look exactly alike. That one of you may have brown hair and one of you may have red hair. Right? So it just depends. Okay. Well, that is pretty much it. So if you have any questions, go ahead and post them in the chat. Again, if you don't think of it right now and you got one later, you can always email me and I'll get to you that way as well. I'm going to go ahead and post the quiz and also the discussion board. The science dailies are also out there, so if you have any questions in the meantime, you can always ask. Alright, so the assignments are posted, so take a look at those, and that's just to keep you updated, make sure everything is jiving with you, that you understand everything, and that way if you don't understand something, you get a question wrong, that would be something you want to ask a question about. I do want to give you guys compliments for doing the discussion boards. You're doing a great job on those. I realize that some sections there is limited participation, so it is difficult to respond to a couple of people if there's only one or two of you working. So just keep doing your best, is all I'm saying. So I really appreciate the work that you are putting into it. All right, I'm going to wait around for any other questions that you might have, and then I will shut her down. Other note, if you haven't checked out, I did post another video from my environmental science class. It's the Adventures of Kentucky Coffee and Sheila Wallaby. You can check that out for a good laugh. It's under the same playlist of Knowledge from the Coffee Cup, as the same thing as these videos. It's pretty comical, I think. Well, alright, I don't see any questions, so you guys must understand the knowledge. So great job, and I'll see you on Thursday.